This is the microphone. Huh? Okay. It's the microphone. Okay, thank you. It's not a drone. Okay. Is everybody here? Please proceed. Okay, we continue to the second session. Marina is here. Where is Marina? Okay. We proceed to the second session that has to do with personalized mycology and vaginal infections. I think that the vaginal infections specifically are, of course, an important topic from the aspect of personalization, having also in mind that a lot of times these vaginal, actually fungal vaginal infections cause quite a lot of distress physically or psychologically, as they cannot be cured very easily. So the first speaker is Professor Marina Randelovic, okay, from Nice, who is going to present us the topic of personalized approach in the vaginal microbiome, the significance of vaginal candidiasis as a cause of vaginal dysbiosis.
Yes. Thank you, Professor, very much. Okay. Dear colleagues, Professor, especially Professor Valentina Arsic-Arsenievich, thank you for having me here today. So as Professor said, I will tell you something about personalized approach to the vaginal microbiome and especially of the significance of vaginal candidiasis as a cause of vaginal dysbiosis. So in recent years, advances in sequencing technologies and bioinformatics have opened new opportunities to explore microbial communities that inhabit human body. These communities are like bacteria, viruses, eukaryotes, or archaea, are collectively referred to as human microbiome. Remarkably, bacterial cells are estimated to outnumber human cells, underscoring the importance of this hidden world. Studies of the human microbiome have revealed that even healthy individuals show significant microbiome variations in gut, skin, and vagina. Much of this diversity is still unexplained, although some factors such as diet, environment, host genetics, and early microbial exposure have all been implicated. So, like a part of human microbiome, very important is the vaginal microbiome, which represents the dynamic and sensitive ecosystem with profound implication for women's health. First investigations about microbes residing in women's vagina date from 1892, when Albert and Gustav Dederlein described rod-shaped gram-positive bacteria, which were later named Dederlein bacilli, and those bacteria were linked to lactic acid production and inhibition of staphylococcal growth. In subsequent decades, it was demonstrated that these bacilli belong to Lactobacillus species, and they are today recognized as the predominant protective members of vaginal microbiota. This is because these bacteria maintain low pH, so below 4.5, in the vaginal environment through lactic acid production combined with hydrogen peroxides and bacteriocides. Also, these bacteria protect vaginal epithelium from microbial invasion and infection, and this is why they have abundantly been associated with overall well-being. The predominance of Lactobacillus species ensures stability and protection, but smaller population of other bacteria and fungi and viruses may be present without causing the disease, because these are part of normal vaginal flora. This balanced ecosystem is dynamic. It is influenced by some factors like hormone status, menstrual cycle, pregnancy, and external factors such as antibiotics or contraceptives. It plays a crucial role in reproductive health, fertility, and protection against infections. With the development of molecular biology, the knowledge about vaginal microbiome is extended thanks to techniques like 16S RNA replicon sequencing, qPCR method, and metagenomics. Building on these methodological advances, Human Microbiome Project was launched as a landmark National Institute of Health initiative. It was conducted between 2007 and 2016 in order to comprehensively characterize microbial communities across the human body and to understand their role in health and disease. It provided reference genomes, baseline datasets, and methodological frameworks that transformed microbiome research. So within this framework, Ravel, with his associates in 2011, categorized the composition of the vaginal bacterial communities in healthy women into 5 community state types by using 16S RNA replicon sequencing. So first community state type 1 is dominated by Lactobacillus crispatus. Second one is characterized by presence of Lactobacillus gasseri, third one by presence of Lactobacillus iners, and fifth one by Lactobacillus jansseni. Only fourth one is characterized by diversity and is not dominated by lactobacilli, but is dominated by facultative and strict anaerobic bacteria like Gardnerella, Atopobium, Prevotella, and Finegoldia. Initial drawbacks of this classification, like sample size and simplification of the bacterial communities to only 5 groups, were addressed by France et al. In 2020 through expansion to 7 groups and 30 sub-CST groups. This framework is validated, but it's not yet standardized. Although CST classification can be useful for statistical analysis, it does not fully capture the biology and functionality of vaginal microbiome. For example, Belgium ISAWA study in 2023 reported that around 10.4% of women with codominance of Lactobacillus crispatus and Lactobacillus iners complicating CST assignment and proposed considering the vaginal microbiota as a whole, rather than using strict CST categories. So this map from the paper Diversity Women and Their Vaginal Microbiota shows world distribution of the vaginal microbiome data studies using 16S RNA replicon sequencing, and it was conducted between 2019 and 2024. As we can see, a lot of studies were done in Asia, majority of studies. But the standardized data sets were provided only from Europe and North America, while other regions of the world remain underrepresented. So this highlights the importance of expanding research worldwide to capture the full diversity and functionality of vaginal microbiome. Disruption of the vaginal microbiome, commonly referred to as dysbiosis, results in loss of acidic pH, reduced protective metabolites, and increased pathogen colonization. Such imbalance can lead to bacterial vaginosis, heightened susceptibility to sexually transmitted infections, and adverse pregnancy outcomes. Understanding its composition and variability is essential for clinical diagnosis and development of some specific therapeutic strategies. So now I'll say something about different vaginal dysbiosis. First, and the most widely spread one is, of course, bacterial vaginosis, which is the most common dysbiosis. It occurs when the protective lactobacilli population is reduced and replaced by overgrowth of anaerobic bacteria. It is followed by symptoms like discharge, odor, and elevated pH. And this state is important because it's associated with increased risk of sexually transmitted infections and adverse pregnancy outcome. Next one is aerobic vaginitis, which is less frequent but clinically important state. In this condition, the protective lactobacilli are replaced with aerobic bacteria such as E. coli or Streptococcus or Enterococcus group. It is followed by symptoms like inflammation, burning, and pain, but is usually asymptomatic. And because of its inflammatory nature, it is linked to infertility and pregnancy complications. So vaginal dysbiosis not only involves bacterial imbalances, but sometimes some viral and parasitic associations can also develop. Dysbiosis can facilitate the persistence of human papillomavirus, human immunodeficiency virus, or other sexual transmitted infections. Altered microbiota can reduce mucosal immunity, creating favorable conditions for pathogens, and this state is important in global epidemiology and reproductive health. Mixed dysbiosis is characterized by coexistence of different dysbiosis like bacterial vaginosis, aerobic vaginitis, or other imbalances. Is characterized by presence of multiple pathogens, which are present simultaneously, complicating both diagnosis and treatment. Diagnostic challenges here are present because of overlapping symptoms and microbiological findings, which are not always straightforward. Lactobacillosis is a rare vaginal dysbiosis and is characterized by excessive growth of lactobacilli. Although lactobacilli are normally protective, their marked predominance can disrupt the vaginal balance. And this state is followed by symptoms such as abundant discharge, discomfort, and irritation. In this state, vaginal pH is usually normal or decreased, and this is why it can be mixed with with vulvovaginal candidiasis. And this is the part I'm going to talk about in more detail. So vulvovaginal candidiasis represents another major form of vaginal dysbiosis and is caused by overgrowth of Candida species. So yeast of the genus Candida colonized the woman vagina in 20 to 30% of all women without clinical symptoms and signs of infections, but by molecular studies, it is determined that this percentage is significantly higher, around 64% are colonized by Candida species. Moreover, as long as the yeast remain in balance with host environment, other members of microbiota and immune system, Candida species are commensal saprophytic organisms. So disturbed balance between yeast and host may occur due to many predisposing factors. We can see them here. So first one is inflammation, then elevated estrogen, diabetes mellitus, some states connected with immunosuppression like HIV infection, chemotherapy on anti-metabolic medication transplantation, also with pregnancy and obesity, and broad spectrum antibiotic usage. Those risk factors can enable yeast to switch from harmless to a commercial or pathogenic form. Symptoms of vulvovaginal candidiasis are itching, irritation, burning, soreness, fissuring redness, vaginal discharge, and dyspareunia. Because of the mentioned risk factor and the very high percentage of women with the colonization of Candida species, it is understandable that 75% of women will undergo at least one episode of vulvovaginal candidiasis. Around 50% will have this state 2 or more times, and what is very important, 8 to 10% of all women will develop recurrent form of vulvovaginal candidiasis, which is defined as at least 3 to 4 episodes of vulvovaginal candidiasis per year. One of the assumptions is that recurrence might result from a higher pathogenic potential of infected species and low susceptibility to antibiotics. It has been shown that Candida species are capable of forming biofilms, and these formations are defined like community of microorganisms which are protected to immune system. They show decreased susceptibility to antimycotics and, which is very important, can spread antimicrobial resistance which make it very important virulence factor. It is well known that Candida albicans is the dominant causative agent of vulvovaginal candidiasis, but in recent years, non-albicans and non-candida species are also recognized as a causative agents of sporadic and recurrent form of vulvovaginal candidiasis. In our work, which is published in 2020, we determined that Candida albicans was causative agent of vulvovaginal candidiasis in 73.8% of cases, Candida glabrata 13.8, cruzi 6.3, Candida tropicalis in 2.2, and also yeast Saccharomyces cerevisiae in 1.9% of cases. So what is the connection between vaginal microbiome and vulvovaginal candidiasis? Vaginal microbiome is thought to inhibit Candida pathogenicity through lactic acid and bacteriocins, hydrogen peroxide, and biosurfactants. Early studies reported that bacteria do not prevent Candida colonization, but rather limits its proliferation and yeast to hyphae transition in Candida albicans. Today, this protective role is debated, since all reduced, elevated, and unchanged lactobacilli colonization patterns have all been observed in patients with vulvovaginal candidiasis. So several studies report that in healthy women, Lactobacillus crispatus from community state type 1 is most evident. And in vulvovaginal candidiasis patients, there's been shown lower overall lactobacilli or shift in species composition, with Lactobacillus gasseri, Lactobacillus iners, and anaerobic bacteria present. On the other way, some culture-based data link Lactobacilli dominance to higher risk for development of vulvovaginal candidiasis, possibly due to lactic acid suppressing immune response, although this was not confirmed by molecular studies. Vulvovaginal candidiasis microbiomes are highly variable, ranging from normal-like to bacterial vaginosis-like communities. Also to mention that antibiotic-induced reduction of lactobacilli may favor Candida, but evidence for a consistent links for this remains limited. So, having outlined the clinical relevance of vulvovaginal candidiasis as a major cause of vaginal dysbiosis, I would now like to present you the results of our work, our paper published in Journal of Fungi, where we aim to examine the pathogenic potential of Candida species isolates by evaluating their ability to form biofilms and their antifungal susceptibility to commonly used antimycotics. By integrating these findings into personalized approach to the vaginal microbiome, we aim to better understand strain-specific variability and its implication for the therapy. So, we examined the biofilm production and antifungal susceptibility for 300 vulvovaginal yeast isolates. Among them, we had 150 Candida albicans strains and 100 non-albicans strains and non-Candida strains. In order to collect yeast isolates, vaginal swabs of 4,569 women with signs and symptoms of infections were observed, and the microbial examination was carried out on each patient in accordance to the standard protocols, included bacteriological, parasitological, and mycological analysis. All strains were cultivated on saburodextrose agar and chromogenic medium and incubated at 37 degrees for 3 to 5 days. The primary differentiation of Candida albicans was done by the color of the colonies, which was green on chromogenic medium, and also based on germ tube test. So all strains were identified using Malditov mass spectrometry. This method was already mentioned here. It's a proteomic analysis. I will just briefly describe the procedure. So we made a T colony layer from Saburodextrose agar on a polished steel target plate. Then we dried at a room temperature and added formic acid and matrix. After drying, it was placed into multi-TOF biotyper Series 1 system, and identification was done by using MBT Compass software. Identification was accepted if the score was above 1.7. Biofilm production assay was done by crystal violet method. Also, briefly, we made the suspension of yeast and then inoculated into microtiter plates, which were previously filled with RPMI medium. After incubation, the content was aspirated, the well was washed with phosphate buffer, and after drying, the crystal violet solution and ethanol were applied. The solution absorbers were read with ELISA reader, and the strains were categorized according biofilm production to non-producers, weak, moderate, and strong biofilm producers. Antifungal susceptibility testing was done in two ways. First one by integral system yeast plus test, which is a commercial test for identification of yeasts, and the other part for determination the susceptibility to 10 most commonly used antimycotics for superficial fungal infection. It was done according to manufacturer instruction, and it was, as I said, screening test for our research. And after we determined susceptibility through integral system yeast test, and after we determined the biofilm producing ability, we did antifungal susceptibility testing by microdilution assay, where we determined minimum inhibitory concentration for fluconazole, clotrimazole, and nystatin as the three most commonly used antimycotics for therapy of vulvovaginal candidiasis. Fluconazole is systemic antimycotic, and nystatin, clotrimazole are topical antimycotics. And we picked 43 strains for this analysis. We firstly prepared antimycotics. We made tenfold dilution. We put it in the microtiter plate. Then we transfer RPMI medium, prepare the yeast suspension and transfer it to the wells, and incubated at 35 degrees for 24 hours. And these are results which we get. So MALDI-TOF mass spectrometry confirmed the 150 strains of Candida albicans. And as for other strains, I will just mention here really briefly maybe some of you know, or all of you, that reclassification of yeasts are undergoing really fastly in recent years. So today, some species which were classified in Candida genus are not today. For example, Candida cruzi, Spichia kudrevsevi, Candida glabratis, Nacaseomyces glabrata, Candida heferis, Pluiveromyces marxianus, Candida lusitaniae, Clavispora lusitaniae, etc. But in order not to confuse clinicians, this is not widely used, so it's much better to still use that previous classification in order to more easy interpretation of the results. So here the species are still presented as Candida species. We determined 62 strains of Candida glabrata, 33 of Candida cruzi, 21 of Candida kefir, 3 Candida lusitania norvegensis, 2 Candida parapsilosis, and among non-Candida species, 26 strains of Saccharomyces cerevisiae. So these are results of biofilm production ability. From 300 strains, 138 had biofilm production ability, which makes them almost half of the isolates that we examined. From Candida albicans strains, 150 of them, even 95, almost 70% had the biofilm production ability. Among them, 18 were strong producers, 28 were moderate, and 49 were weak producers. Among 124 non-albican species, around 30% were biofilm producers, 5 strong, 12 moderate, and 19 weak. And among 26 strains of Saccharomyces cerevisiae, 4 were moderate and 3 were weak biofilm producers. This is very important because all of those isolates which were biofilm producers has a very powerful virulence factor to provoke recurrence and chronicity of vulvovaginal candidiasis. Also, what can influence the chronicity and recurrence is resistance or low susceptibility to antimycotics. So these are results of antifungal susceptibility testing to nystatin, fluconazole, and clotrimazole by using microdilution method. So for fluconazole was easy because we have breakpoints which are defined by UCAST and CLSI. So we determined that around 94% of strains were resistant of Candida albicans, were resistant to fluconazole, and 20% of Candida glabrata, 50% of Candida cruzi, and all strains of Candida kefir. It was more difficult for clotrimazole and nystatin because for topical antimycotics we don't have defined minimum inhibitory concentrations from UCAST and TLSC, so we try to find it in the literature. It's really hard to define those, really difficult to define those breakpoints. So we took the breakpoints from Richter in 2005, who used 1 microgram per milliliter as a breakpoint. According to Richter, so all of the strains were resistant to nystatin, and clotrimazole was the best option here because all of the strains were sensitive to clotrimazole, but 33%, around a third, strains of Candida glabrata which were resistant. Here we presented the biofilm producers of different yeast species with high MIC values. So these isolates at the same time produce biofilm and show low susceptibility to antimycotics. Among Candida albicans strains, 20% of biofilm producers show low susceptibility to fluconazole. Among non-albicans Candida species, 4%. When we look at nystatin, 20.7% of Candida albicans biofilm producers were resistant to nystatin, 14.5% of non-albican species, and 23.1% of Saccharomyces cerevisiae. Only 4% of non-albicans Candida species, as I said, Candida glabrata, were resistant to clotrimazole. And when we observe total isolates, so 7.7% of total isolates, biofilm producers, were resistant to fluconazole, 14.3% to nystatin, and 1.7% to clotrimazole. 1% of all examined strains were at the same time biofilm producers and resistant to all 3 antimycotics at the same time. So, based on our findings, as well as numerous reports in the literature, we can conclude the resistance of yeast isolates to commonly used antifungal agents represent a major factor which contributes to the treatment failure and relapse of vulvovaginal candidiasis. In response to this growing challenge, novel therapeutic agents such as oteseconazole, which is a selective oral inhibitor of fungal lanosterol demethylase, and ibrexafungerp, which has a similar mechanism of action like echinocandins, have emerged as a promising future options. These agents may offer improved efficacy, particularly in cases with ulcer-resistant infections. However, antifungal resistance alone does not fully explain the persistence and recurrence of infection. As we already said, our results highlighted the importance of biofilm production as the key virulence factors and key influence factors for the therapeutic outcome. This is because biofilm significantly enhances the resilience of sessile candida cells and limits antimycotic penetration, promoting chronic or recurrent infection. Consequently, even in the presence of newer antifungal agents, Candida biofilm still remain the major barrier to successful eradication and contribute to persistent infection. At this point, it becomes evident that vulvovaginal candidiasis cannot be adequately managed with uniform therapeutic strategy. Although traditionally considered a simple infection, this is in fact highly heterogeneous condition. Clinical presentation, recurrence, and response to therapy vary widely among patients to different Candida species, but not only that, also antifungal resistance, biofilm forming capacity, and vaginal microbiome. Our research demonstrated that this factor can directly influence the treatment outcome. And consequently, empirical approach is insufficient, particularly recurrent infection, clearly illustrating why one therapy does not fit all patients. It is also very important that therapeutic success is influenced not only by antifungal choice, but also by the microbial environment in which treatment is applied. Antifungals may effectively eradicate planktonic Candida cells, but leaving biofilm reservoirs, leaving those sessile cells in biofilm, especially in these biotic conditions. Moreover, antifungal treatment alone does not restore microbiome balance and may even contribute to recurrence. This is why the vaginal microbiome shapes the initial disease course and determines the long-term outcome therapy. In this context, personalized approach to the vaginal microbiome becomes essential. Integrating species-level identification, antifungal susceptibility testing, and biofilm detection into routine, at least for recurrent vulvovaginal candidiasis patients, would enable therapy to be tailored to the individual patient rather than relying on uniform regimens. This approach is particularly relevant given the increase of non-albicans Candida species and Saccharomyces cerevisiae as the causative agents of these infections. Looking forward, future researchers should not focus only on the development of new antifungal drugs, but also, as we mentioned previously, on natural products and microbiome modulating interventions, like usage of probiotics, prebiotics, or even vaginal microbiome transplants. Such strategies support personalized medicine and aim to restore microbial balance while targeting fungal resistance. This perspective underscores that vulvovaginal candidiasis is not merely an isolated fungal infection, but is a significant contributor to vaginal dysbiosis. Therefore, effective management requires individual diagnostic and therapeutic approach, which address both the pathogen and the vaginal ecosystem, with the ultimate goal of improving outcomes and reducing recurrence. Before I finish, I just want to send warm regards from Professor Susan Otasevich from Niš and to send her invitation to JOY special issue, Diagnosis and Management of Human Mold Infection, second edition in Journal of Fungi, and all participants of this workshop will have 20% discount if they contribute to the special issue. So thank you very much.
Thank you so much for your full-scale presentation and also the presentation of your interesting scientific work. Also, thank you for the offer for the special issue. Okay, I think we should follow the approach of the previous sessions, so possibly keep the questions for the end of the session. And we continue to the second presentation that is by Dr. Slavika Dastis. Yes, am I spelling it correctly? Okay.
Yes, correctly.
And so it concerns the personalized approach in detection and understanding of vaginal infection by applying yeast quantification and biofilm production. Okay.
Well, thank you very much. My colleague is Slavica Dacic, medical doctor, and I am Ljiljana Pavlović, also medical doctor. and today I am going to present
Okay.
this lecture. Dear colleagues, as mentioned before, now I'm going to give a small presentation regarding a personalized approach to the detection and understanding of vaginal infections through yeast quantification and biofilm production assessment. Okay. As Professor Marina already emphasized, and I will repeat some notes that she has already said, vulvovaginal candidiasis is a fungal infection of the vagina and external genital area caused by the fungi of the genus Candida, most commonly Candida albicans. After bacterial vaginosis, it is the second most common cause of vaginitis, between 15 and 30%. Vulvovaginal candidiasis has a high incidence and prevalence, making it a major public health concern. It occurs at least once during a woman's lifetime of 70 to 70% of all women, while in 55% of women, the infection recurs multiple times during lifetime. It is estimated that 5 to 9 of women have recurrent vulvovaginal candidiasis. It is not subjected to mandatory reporting of infection diseases. It is not classified as a sexually transmitted disease. According to the CDC Center for Disease Control and Prevention, a vulvovaginal candidiasis can be classified as uncomplicated and complicated issue. Among Candida species, 75 to 90% of isolates belong to Candida albicans. Then between 10 and 25% of isolates belong to non-Candida albicans Candida species, including Candida glabrata, Candida parapsilosis, Candida tropicalis, and Candida krusei. Vaginitis caused by non-albicans Candida species is clinically similar to vaginitis caused by Candida albicans. However, non-albicans Candida isolates are often resistant to antifungal therapy, as we already have heard. Most common risk factors include hormone therapy, pregnancy, immunosuppression, antibiotic therapy, diabetes mellitus condition, and genetic predisposition. On this slide, you will see in Serbian. So on the next slide, it is also the same in English. Microbiological diagnosis starts with taking a vulvovaginal swab. Then we do microscopic examination to see if there is evidence of the presence of blastoconidia, pseudohyphae, and hyphae. Then we are trying to culture examination on saburodextrose agar or chromogenic media. After that, we can use identification of identification methods, and there is a germination tube test, Microbiomic production test, and chromogenic media. For first and second test, we can, by these tests, if we use them, we can do the identification of Candida albicans species, and chromogenic medium can be used to differentiate between Candida albicans, Candida tropicalis, and Candida krusei species. Identification of other yeast species I've already have said. We can use biochemical tests, multi-top mass spectrometry, or molecular methods. And in the end, antifungal drug susceptibility testing can be done with 3 methods: diffusion method, dilution method, and using commercial media. The diagnosis of vulvovaginal candidiasis requires correlation between symptoms, clinical findings, microscopy, and culture results. Symptoms of vulvovaginal candidiasis are not specific. The most common symptoms is itching, but its predictive value is accurate in only about 38 of patients. Measurements of vaginal secretion pH is important but insufficient for establishing the diagnosis. If its pH value is above 4.5, it may indicate bacterial vaginosis or trichomoniasis. Direct microscopic examination is mandatory, but false negative results may occur in up to 50% of cases. It is also useful in the diagnosis of bacterial vaginosis and trichomoniasis. An increased number of leukocytes may indicate a mixed infection. Culture is required in cases of symptomatic vaginitis with negative microscopic findings for fungi, especially in patients with complicated vulvovaginal candidiasis. It enables antifungal susceptibility testing and assessment of biofilm production capacity. Isolation and identification of yeasts in the absence of symptoms is not an indication for treatment. Many women with vulvovaginal symptoms receive an incorrect diagnosis or inadequate treatment because most do not have laboratory confirmation of the causative agent. Which are the problems? Standard microbiological diagnostics are often insufficient because they do not distinguish between infection and colonization. Use of empirical therapy without antifungal susceptibility testing, and treatment failure and recurrence. What is the solution? A personalized approach to the diagnosis of vulvovaginal candidiasis. Why is a personalized approach important in the diagnosis of vulvovaginal candidiasis? A personalized diagnostic approach is important because each woman has a unique vaginal microbe that is subject to change. As Professor Marina already said, there is no one solution for every woman. Key reasons include accurate distinction between infection and colonization. Not all detection of Candida indicates disease. Some women are asymptomatic carriers. Personalized assessment helps avoid overdiagnosis. Identification of different Candida species. Non-albicans species often show different clinical behavior and high resistance to standard antifungal therapy. Improved treatment selection. Empirical therapy may fail, especially in recurrent or complicated cases. Individualized diagnostics, including culture and susceptibility testing, guide more effective therapy. Reduction of recurrence. Recurrent infections often result from inadequate initial diagnosis or inappropriate treatment, which personalized diagnostics can help prevent. Better clinical correlation. Combination of symptoms, clinical findings, microscopy, pH values, and microbiology ensures a more reliable diagnosis than any single method alone. Important steps in the personalized diagnostics of vulvovaginal candidiasis. Detailed clinical history, symptoms, recurrence, risk factors. Clinical examination and assessment of vulvar and vaginal findings. Measurement of vaginal pH values. Microscopic examination of vaginal secretion. Microbiological culture and species identification, antifungal susceptibility testing when indicated, assessment on biofilm production in recurrent or complicated cases, differentiation between infection and colonization, and integration of clinical, microscopic, and laboratory findings for final diagnosis. Microscopic examination of vaginal secretion, what should we report? Presence of epithelial cells, number of morphotypes of microorganisms according to Nugent score or Hay-Eisen grading system, presence of leukocytes, and presence of blastospores, pseudohyphae, and hyphae. Microbiological culture and species identification, it requires cultivation on Saburrodextrus agar or homogenic Candida agar. Identification is performed using the germ tube tests, biochemical tests, the Vitek2 system, and Malditov mass spectrometry. Also, I will show you on the next slide application of homogenic media, germination tests, and examination of blastoconidia morphology, differentiation and identification of isolates using Malditov MS, examination of biofilm production, classification of Candida albicans, non-albicans, and non-Candida strains according to their ability to produce biofilms, as we also have heard. There are strong biofilm producers, moderate weak biofilm producers, and non-producing strains. Quantification of growth. We should do the cultivation of Sabura dextrose agar or chromogenic Candida agar. There are semi-quantitative methods and quantitative methods. Clinical significance. Differentiation between colonization and infection. Monitoring of therapeutic response. What are the advantages? Objectification of findings. Colony forming units per milliliter indicates the density of Candida population in the vagina. The number of cells, colony forming units per milliliter, also influence the intensity of quorum sensing. And what is quorum sensing? It is a mechanism of chemical communication between Candida cells through signaling molecules such as farnesol or tyrosol. When the concentration of signaling molecules exceed the threshold, quorum sensing is activated. Main effects include regulation of growth, control of the transition to the hyphal form, and coordination of virulence gene expression. Previously, colony density was categorized as sparse, moderate, heavy, or very heavy based on the number of colonies observed on the agar surface. The main limitation was the subjectivity of the assessment. Number of colonies per sample. We can use criteria of Higa-Schrieder and colleagues recommended by Suarez and Lancha for the quantification of vaginal fungal culture. If the colony forming units are below 10 of the primary isolate, it is indicator of asymptomatic vaginal fungal colonization. If the colony forming units are above 10 of the primary isolate, it is an indicator of symptomatic vulvovaginal candidiasis. According to practical guidelines, actually UK mycosis guidelines, reporting is based on the number of colonies on a standard plate, direct inoculation from a swab. Light growth means less than 10 colonies per plate. Between 10 and 99 colonies, it means moderate growth, and heavy growth if there are more than 100 colonies per plate. Quantitative microbiological culture. Vaginal samples are suspended in 1 milliliter of sterile saline solution, vortex and serially diluted. Aliquots are inoculated onto Sabura dextrose agar and incubated at 35 to 37 Celsius degrees for 24 to 48 hours. Colonies are counted and results are expressed as colony forming units per milliliter. Calculation actually is the following. Colony forming units per milliliter is equal to number of colonies multiplied with the dilution factor and divided with inoculation volume. This quantitative approach enables more precise determination of fungal burden. If a fungal vaginal culture shows a moderate or high number of Candida colonies and the patient has typical symptoms, itching, burning, cottage cheese-like discharge, a fungal infection is present and treatment is indicated. What are the barriers and the limitations to the use of quantification in laboratory practice? First of all is methodological variability. Differences in sampling, dilution techniques, and inoculation volumes can affect colony forming units per milliliter results and reduce comparability between laboratories. There are pre-analytical factors. Improper sample collection, transport, or storage may lead to underestimation or overestimation of fungal burden. Biological variability of the vaginal microbiome. The vaginal microbiome environment is dynamic, so Candida levels may fluctuate independently of symptoms. Difficulty distinguishing colonization from infection. Even high colony forming units counts do not always correlate with clinical disease, complicating interpretation. Lack of standardized cutoff values across laboratories. Although some guidelines exist, universally accepted thresholds for clinical decision-making are still limited. Time and resource requirements. Quantitative culture is more labor-intensive, time-consuming, and costly compared to qualitative methods. Influence of non-albicans Candida species. Different species, as we already see, in varying growth rates and virulence, which can affect quantitative interpretation. Limited integration into routine diagnostics. Many clinical laboratories still rely on conventional qualitative cultures or microscopy, reducing widespread adoption. Overall, while quantitative methods improve objectivity, their clinical utility depends on the standardization and careful correlation with clinical findings in vulvovaginal candidiasis. I should emphasize a note. In rapid tests for the diagnosis of vaginal candidiasis, a cutoff value of 10 on 4 coloniform units per milliliter for Candida albicans has been used. Therefore, in most studies, this value represents an approximate threshold between low and moderate to high colony counts. Now I will describe two scenarios. Scenario 1, low colony forming units counts plus presence of symptoms, itching, burning, thick white discharge, and redness. A possible early stage of infection or low-grade candidiasis when fungi have not yet formed heavy colonization but have already triggered an inflammatory response. This situation is common in sensitive patients or in those with an altered vaginal flora, for example, after antibiotic use. In some women, even small amounts of Candida can cause intense symptoms due to local hypersensitivity or a disrupted vaginal immune barrier. A possible fungal infection caused by non-albican species, for example, Candida glabrata, which can produce subapical symptoms with poor growth in culture but may be treatment resistant. Identification of the Candida species is essential, and antifungal susceptibility testing, antifungigram or antimicrogram should be performed whenever possible. Scenario 2. High colony forming units, but the patient has no symptoms. This represents colonization or asymptomatic carriage. Quantification is a tool, but is not by itself proof of disease without clinical correlation and microscopy. Each finding should be interpreted in the context of symptoms, vaginal pH values, and the overall condition of the patient. So, conclusions are, there is not always a direct correlation between colony-forming units count and symptom intensity. A low colony-forming units count is not always indicative of colonization. It can also be present in infection, and vice versa, a high colony-forming unit does not always indicate infection. And for the next few slides, I will show you something about biofilm formation. Candida species causing vulvovaginal candidiasis can form biofilms, which are structured communities of cells embedded in an extracellular matrix. Biofilm formation is an important virulence factor because it increases resistance to antifungal therapy, protects fungal cells from host immune responses, promotes persistent and recurrent infections, and makes eradication of infection more difficult compared to planktonic free-floating cells. Assessment of biofilm production is therefore relevant in recurrent and complicated cases as it can influence treatment choice and clinical outcome. Assessment of biofilm production capacity has diagnostic significance, assessment on infection severity, and therapeutic significance, selection of antifungal strategy. Determining the ability to produce biofilm in correlation with the clinical presentation and symptoms is an important criteria for distinguishing colonization from infection. Methods for biofilm detection. In vitro, we can use microtittle plate assay with crystal violet as a crystal violet assay and XTT reduction assay. Microscopy using scanning electronic microscopy and molecular methods analyzing of expression of the biofilm specific genes. And on the next slide, I will show you assessment of biofilm production capacity in Serbia is performed using the crystal violet staining methods proposed by Stefanovic and colleagues adapted for Candida species. Preparation of yeast suspension with a turbidity of 0.5 McFarland's units in saline, inoculation of the yeast suspension on RPA media into microtiter plates, washing with phosphate buffer after incubation, and adding a solution of crystal violet dye and ethanol. Reading the results after transferring the solution to a new microtiter plate using the ELISA reader, and classification of strains based on their ability to produce into strong, moderate, weak producers and non-producers. Absorbance is measured at 595 nanometers. And how is cutoff defined? Actually, cutoff values is optic density of negative control plus 3 standard deviation. And criteria for interpretation of biofilm productions are the following. If the optic density is lower than a cutoff value, there is no production. If the optic density is between 1 and 2 cutoff values, it is weak production. If the optic density is between 2 and 4 cutoff values, it is moderate production, and if it is above 4 cutoff values, it means strong production. A combined and personalized approach enables the definitions of clinical microbiological scores whose role is differentiation between infection and colonization, as well as assessment of infection severity, identification of high-risk patients, individualization of therapy leading to better clinical outcomes, and reduction of recurrence rates. Low colony forming units plus no biofilm production means colonization. High colony forming units plus biofilm production mean active infection. Limitations of quantification methods and biofilm production assessment. There are lack of standardization of methods, higher cost of analysis, and complexity of diagnostics. And in the conclusion, quantification of yeast from vaginal samples together with assessment of biofilm production capacity represents a key component of personalized laboratory diagnostics of vulvovaginal candidiasis. This combined approach improves diagnostic accuracy, supports differentiation between colonization and infection, enables better risk certification, and contributes to more effective individualized treatment and reduction of resource rates. And I would like again to emphasize, as you have heard yesterday, finally, as a part of this FCF project idea, a mobile application was developed to help patients with the diagnosis of vulvovaginal candidiasis. To answers to questions about symptoms and the quality of vaginal discharge, patients are guided toward the diagnosis. At the end, I would like to thank you, my colleagues, Dr. Slavica Dacic, who offered me to present this lecture, and Professor Valentina Arsic-Arsenijevic, who also invited me to this session. Thank you very much.
Thank you very much for this extended and more technical approach to the personalization of vaginal candida infections. We shall proceed to the next one. As I said previously, questions could be kept for the end of the session. So the next one will be made by Dr. Rajendra Durisic. Yes, okay. It concerns candida infections in pregnancy. So a systematic review of the literature and single-center data from Serbia. So
yes.
Please.
Thank you. Thank you. Should I open the presentation? Okay. Okay, dear colleagues, it is very, very nice to be today with you. My theme today is Candida in pregnancy. It was actually an invitation from a global perspective to observe the global ambience and momentary informations we have. So it was very deficient in information about Candida in pregnancy, especially considering the risk factors than the standardized methods we use for testing and for diagnostic methods. So it was a little bit challenging to put all that in one standardized manner. And considering there was no previous systematic review with the PRISMA method that observed all those informations in one systematic review. This was the first approach with this PRISMA method that succeeded to observe global perspective as well as single-centered Serbian data. So first of all, vaginal candidiasis, as you know, is very much it has a big prevalence in population. First of all, it is 10% of all women in generative age has in some cases this problem. But in pregnancy, it is up to 30%. So we can all see that is a global problem. Especially non-albicans Candida species are getting much and much more, as you see next to albicans Candida, it is much and much more, as you say, in the population, because we have much better methods to diagnose those species. So that could be also the reason for the appearing more and more recurrent Candida infections. Also, that recurrence is unclear because Candida has its virulence factors that are still unfamiliar to the full picture to us. Also, as we said, procedures lack standardization. As we will see in the future slides, those procedures are very variant, and that was the difficult part of this research because it was very hard to make standardized methods to diagnose and to treat Candida. So this was a big invitation for this approach and this research to standardize and to have that calling to standardize the procedures of testing, diagnose, and also the risk factors that predispose women with this problem. So the methods we used was the PRISMA protocol that was actually a statistical, analytical method to observe the global research we had. And as you can see on this figure, on the upper left corner, we used three main electronic literatures from the three main sources. It was Medline, Web of Science, and Scopus. So we observed 350 research papers, but we needed, of course, to exclude some of them because they were not eligible for our research. So, as you can see, we concluded that 36 research papers were eligible and approved for our research. It was, of course, research papers considering all the continents. Next, what we wanted to observe is the national point of view. So we used the second method with the Serbian National Referential Laboratory for Medical Mycology. So it was a center place to investigate the previous results and specimens from the national patients. We excluded some of the research papers that were done on animals, then some of the papers that were abstracts, and papers, of course, that were treated with that treated Candida before and after, because of course you could get false positives or false negatives. So those were all excluded. And we managed to get all the necessary review papers globally. So sample size was actually quite a big number, because we estimated it was about 25,000 pregnant women, approximately between 11 and 64 age gap. And of course, in Serbia, it was plus 700, because it was our data from the national point of view. And what we managed to see here is, as you will see in the future slides, it was approximately between 15 and 55% of women get candida in their lifetime, depending on the country and the continent. Whilst in Serbia, we found about 48 positive candida species. As you will see, albicans is in about 77%, and that is important to know that in recurrent forms, Candida albicans is a little bit less presented comparing to non-albicans species. Because, as you can tell, the non-albicans species make recurrent forms of Candida, such as Candida krusei, Candida tropicalis, and Candida glabrata. This is an example from our research paper. We actually managed to get about, as you saw, 25,000 pregnant women considered in Africa. As you can see, we had few variables from risk factors to clinical symptoms and diagnostic methods that were used. And as you can see, though in the column of risk factors to the on the right side of the screen, we can see immunocompromising state is the big factor that induces Candida colonization and invasion. Next is using prolonged antibiotic use, gestational period, of course, and amongst clinical symptoms, you see pruritus, discharge, burning sensations, and etc. Our biggest part of the research and the biggest population considered was in America, because there was about 15,000 specimens in positive pregnant women. But on the other hand, we have, as you can see, Africa, Europe, and I will show you, of course, the other ones. As you can see, the methods to diagnose is very variable, but those were the mostly, Candida was mostly cultivated on chrome agar. Also, some proteomics methods like MALDI-TOF, that is now the most preferred method, and that should be worked on. Then what should you see? Maybe the map view will be more precise for you to understand and observe the clinical perspective. That basically globally we see between 15 and 55% prevalence in Candida species, but even up to 65% in diabetic populations. And as you can see, the South America, America is a little bit more, as you can see, prevalent with the Candida species. And those were all the triggering factors for us to make this our topic and make a research about it, because there was no before systematic PRISM oriented review. Of course, the causative agents were one of the most important objectives of this study, because as you can see, non-albicans Candida are very much prone to make recurrent forms of Candida, and that is the big problem and one of the main reasons why we should make standardized protocols for diagnostics to fulfill and to reduce the rates we saw at the beginning. As you can see, the national data was observed in 1 decade from 2013 to 2023. And as you can see, we observed some of the variables on this table. We've had compared the vulvovaginal candidiasis and recurrent form of vulvovaginal candidiasis. As you can see, the Candida albicans is much more, in much more bigger percentage abundant in vulvovaginal form. Whilst in recurrent forms, there are non-albicans Candida, such as Candida krusei, Candida tropicalis, Candida glabrata. You can all see that in our table. Then there was fluconazole sensitivity. Of course, the Candida was much more sensitive, and the vulvovaginal candidiasis compared to recurrent forms is much more sensitive than the non-albicans Candida. We considered symptoms, which are quite important because you can, by the clinical perspective, provide and see which kind of candidiasis it could be. So, for example, malodor, as you can see, is much more abundant in recurrent forms. So, all that raises a suspicion that if a patient has that malodor, that it could be recurrent form. Also, signs, as you can see, redness, edema, mucosal irritation, all those were considered and made to notice. Risk factors. Risk factors, of course, diabetes mellitus, which significantly raises the risk for pregnant women to have Candida. Next, antibacterial drugs with their prolonged use, immunosuppressive drugs, and contraception usage. And what is here important also, on the right part of the screen, we have the presentation of total numbers of cases Candida versus Candida infections versus recurrent infections. And you can tell that there is a total number approximately between 40 and 80% through those years considered and investigated, compared to Candida infections, about 30 to 40%, and recurrent infections are about 20%. Of course, since this is the study that was considering global as well as national information, as you can see, studies we use on the left from the global perspective all had to be quality assessed. So there was many variables that were that had to be fulfilled in order to investigate those studies, such as such as a detailed setting description, valid or reliable exposure, objective standard measurement and criteria, etc. And at the end, we get a quality score that should be exactly as presented in order to be in our research paper. Exactly as we saw, we had from 350 studies, we had only 36 at the end, because those were the rigorous study methods and analytical analysis. So we managed to have 25,000 pregnant women that revealed prevalence of Candida, ranging from 70 to up to 59% prevalence, even more with diabetic patients. And in Serbian registry, it was indicated about 48% of occurrence in vulvovaginal candidiasis during pregnancy. And those were all quite important because we need to invest in our standardization of all these methods in order to have one global perspective and one standardized approach to each patient in order to reduce all percentage we saw on the screen. On the conclusion, there was significant variability across all the countries and all the continents. Exactly, that is why we do this as we do it. We need to develop all the strategic methods to make it more standardized and more alike one to each other in order to reduce the prevalence. Therefore, it is necessary to develop a valid and standardized sampling methods linked to a platform for patient risk assessment. This standardized methodology and approved screening tests and guidelines are important to comprehensive comparative laboratory analysis during pregnancy. And to improve our understanding of fungal prevalence and resistance patterns, we created exactly the fungal case finder to increase this effectiveness in diagnosis and treatment on fungal infections in the future, including vulvovaginal candidiasis in pregnant women. Thank you.